The samples come in. They can be blood samples. The samples come in, and all of those samples contain cells, cells that make up the body, and all of the cells contain DNA.
Yeah, I know, recording. Right, so sorry. So you're getting the sample at this point in time. You're getting the samples in. You're cutting them down, or they've all contained cells. So is this the first stage of trying to get the cells out?
What we have to do is break open the cells so that the DNA is released, and then we have to purify it. And the key thing here is that all of the cells within one person have the same DNA.
Right.
So if I looked at your hair roots or your blood or your skin, your DNA would always be the same. Whereas the DNA from different people is different.
So so how do you break open the cells?
Well, quite simply, with a detergent, it sort of bursts the cells. And there's something called an enzyme in there as well, which helps with that process. The cells just burst open, everything comes out, so there's DNA plus lots of other things in the cells. And then we have to purify the DNA away from the other cellular contents.
And you still do it at this part of at this stage?
Yes, that's what's happening right now. So each sample is in a tube.
Right.
Each tube has a unique number, identification number on it.
Right.
And it's really a series of extractions. You add a chemical, mix the tube, and then spin it in a centrifuge to separate out. And then each time you're taking the fraction that has the DNA in it, and it gets more and more purified as you go through.
So, I only did O-level biology, so I mean, DNA is in the nucleus. It's the stuff around. What do you call that stuff around the nucleus?
Cytoplasm.
I knew plasm wasn't it, so.
Shall I cut it?
Okay.
By adding ethanol. And what happens is the DNA comes out as solute.
No.
Yeah.
Running.
So, what stage of the process are we at here?
What we're doing now, this is the phenol extraction that's happening at the moment.
Yeah.
So the DNA, the solution has been split into two layers, two phases.
Yeah.
Okay. On the top, you've got the aqueous phase, which is mainly water, and the DNA is in there. On the bottom, you've got the phenol phase, and most of the protein and other things are in there. So you just take this off,
And discard
and the
the
DNA is
rest.
there, and just discard the rest.
And then that's got what, maybe 2 more passes, or
Well, after that, it goes through a couple of passes of ethanol precipitation to clean it up even further. So he's carefully just taking off the top layer, which has the DNA in it.
what sort of training, what sort of qualifications are you talking about for people to do this job?
The staff are all either graduates in a biological science or people who have an HND court in terms of chain of custody. The samples have to be brought here by a policeman. They have to be handed over to the staff here and signed for. Whereas in civil cases, we usually get most of the samples in the post.
I heard a story earlier this week, I don't know, it might just be apocryphal, but about this guy in Scotland who was jailed for 3 years for incest with his daughter between the ages of 16 and 19. Sorry, he was jailed for 3 years. Then he was let out, and then he recommitted it. And they were about to jail him again, and some sort of test was done, a DNA test or something, and it found out that his daughter wasn't his daughter after all. The wife had been fooling around with a milkman, and he had his conviction quashed. Is that apocryphal, or would that sort of thing actually happen?
I'm not familiar with that particular case myself, but I have seen a similar case where the charge was incest. And another important element of this is obviously paperwork.
Yeah.
We have an unbroken chain of paperwork from the samples being taken by the physician to coming in here and going right through the process. So, for instance, when samples are taken, the physician fills out a set of paperwork, and the individuals being sampled have brought photographs. So he has to sign that this is a good likeness of the person, so that people can't sort of switch and send their brothers and whatever. So we receive this paperwork with the blood samples, and then we have a form for each case. Each case is given its own specific number.
What about is this confidential work?
Yes.
I assume that maybe in a paternity case is not as important, but would the